Hello there. Thank you for joining us today. My name is Edward Hahn. I'm a product manager at Bruker Cellular Analysis, and today I'll be presenting single cell technology to revolutionize functional cell screening and biomarker development. I'm presenting this in collaboration with Rhino Bio, who is our distributor within the Korea region. So, quick introduction to our instruments, our IsoSpark and IsoLight instruments. We have over 277 worldwide placements, and we're in 100% of the top 15 pharma, 87% of comprehensive cancer centers, and our clients range from biopharma to academic to CROs and distributors. So, with all these instruments, we've been able to achieve over 140 publications over the span of the last decade. So, where exactly does our instrument fall in the place of cellular biology? Well, I always like to start with this particular timeline here, where if we look at the 1980s to the early 2000s, the major breakthroughs in cellular biology were being done through surface marker analysis. In other words, typically through flow cytometry. So, during this phase of cellular biology, everyone was exploring different types of surface markers and understanding more and more surface markers to define their cellular subsets. However, we have a saying called don't judge a book by its cover. So just judging these cells by their surface markers does not give you a full and complete understanding of cellular biology. As we entered into the 2000s and the early 2020s, we had a lot of different types of genome breakthroughs. So sequencing and genomics was becoming cheaper and cheaper during these decades, and that allowed many labs to gain access to understanding the transcriptome of these cells. Again, though, the transcriptome does not paint the full picture of what these cells are capable of. The RNA and the DNA inside of these cells only really colors in the potential of what these cells can do. Rather, what our company is focused on and where we think the future of cellular biology is in functional breakthroughs, where we're not just characterizing the surface markers or characterizing the genomics, we're now characterizing the actual functional performance of our cells. And this functional performance is essential to gain better biological understanding that's driving a lot of new and innovative therapeutics, such as CAR T, TCR-T, or TIL therapy. And these functional breakthroughs are extremely important to characterizing the cellular phenotypes in these therapies. So Bruker Cellular Analysis instruments really specialize at measuring cellular functionality and reveal the insights needed in order to advance biological research. Um, so with regards to the main type of functional modality that the IsoSpark and IsoLight measure, it's functional cytokine secretion. Functional cytokine secretion is, of course, very important for a wide variety of different immune cells. In the context of cancer immunotherapy, it's particularly important since functional cytokine secretion is what allows cells to enact biological outcomes, such as killing cancer cells, or communicate information by recruiting other types of immune cells to the site of cancer. So, again, functional cytokine secretion is a very essential part of understanding what these cells are doing and how they're behaving. So the ISO platform really allows you to gain access to understanding these cells and to unlock different types of proteomic insights. So what we have is a wide range of different cell types and different types of protocols that we support. And these cells are processed and placed into our barcoded chips, and that allows you to reduce the amount of hands-on time that you're really using to analyze your cells. These chips are then placed into our IsoLight or IsoSpark instrument, where they're automatically processed in order to generate different types of functional proteomic insights. And finally, all of this data is fully processed in our IsoSpeak software, where analytics are performed and they generate full visualizations that can be easily included in any sort of grant or publication that you're looking for. So looking through at our different chip types, we have a wide variety of different chips to suit your needs. For starting off at the top, we have our single cell secretome chip, which detects protein secretions from cells at a single cell level. We also have our automated bulk protein analysis platform that helps you to save time and money to analyze any types of biofluids that you might be interested in. And finally, we also have an intracellular signaling platform where we're able to assess multiple different phosphoproteins from the same individual single cells. So for today's presentation, we'll be focusing on the first chip, the single cell secretome chip, which again detects protein secretions from cells at a single cell level. So the way that this particular chip works is you first load in your sample with your cells, and the design of the chip contains thousands of these tiny microchambers where these cells become situated in. So you'll see here from this video that you have individual cells inside of these microchambers where they're then allowed to secrete their cytokines. And these cytokines are captured on a proteomic barcode, which is basically an array of pre-patterned capture antibodies on each individual chip. And this is then automatically processed by the IsoSpark or IsoLight in order to generate the signal that is then imported into our software, IsoSpeak. So here's a quick view of the technology itself. You have your IsoCode chip, again, with individual microchambers in order to capture single cells. Looking at all of the different analytes that we can cover, we have multiple different panels available. You can capture up to 30 plus cytokines and analyze the secretions of 30 plus cytokines from each individual single cell in your sample. So looking through at the different types of cytokines you can look at, there's effector, chemoattractive, inflammatory, stimulatory, and regulatory functions that you can assess at the same time for each individual single cell. We have a variety of different panels available for you. Please feel free to contact RhinoBio or Bruker Cellular Analysis representative in order to learn more. So common question is, why exactly do I need this platform? I already have different types of proteomic platforms such as flow cytometry or bulk ELISAs that I can use to analyze my cells. So looking through there, one of the key aspects to really keep in mind is that certain specific cells are polyfunctional, meaning that they're capable of secreting multiple cytokine types at once. Polyfunctional cells are extremely important for a wide variety of different types of applications. So let's go over a quick example here. Let's say you have this small collection of cells, and your goal is to identify the polyfunctional cell and whether a polyfunctional cell is present. Well, if you utilize bulk analysis, you look at the average of all secretions, but you don't know which individual cytokines come from which cells. So you're not able to fully assess whether a specific population has polyfunctional cells or not. Similarly, for flow or mass cytometry, what you're really doing is you're looking at either surface markers or intracellular markers, and that again does not give you the full picture in terms of the actual functionality of these specific cells. Instead, what our platform offers is single cell analysis through the ability to look at each individual single cell and identify their functional secretions. So by separating these individual cells into the microchambers like we do on our chip, we can now identify the polyfunctional cells by seeing that specific cells are secreting multiple different cytokines at once, which really differentiates them from other cells that may only be capable of secreting 0 or 1 cytokine at once. So comparing to different technologies, again, bulk ELISA, you're averaging all secretions and you lack that single cell resolution. With flow cytometry or mass cytometry, you're looking at surface or intracellular markers, but that fails to really reveal cellular functionality. It fails to capture these cells in action. And there are some platforms that are more focused on cellular secretion, such as ELISpot and FluoroSpot, but these platforms are extremely limited in their multiplexing capabilities, so they can only typically do 1 to 4 cytokines at once, whereas our IsoSpark platform can allow you to look at 30 plus cytokines at once to really gain a large depth of understanding of your single cells. And the way that we then process this information is we utilize this unique metric that we generate called the polyfunctional strength index to give a critical view of cellular potency. And the way that polyfunctional strength index is calculated is we start by characterizing the percent polyfunctionality in your population. So what percentage of the cells that you've loaded are capable of secreting 2 or more cytokines at once? We also characterize, of course, the cytokine secretion intensity. So how much cytokine is being secreted by those cells that are secreting multiple cytokines at once. So by combining these two metrics together, we've generated a unique type of metric called the polyfunctional strength index to really give a very quick view of potency for your sample. So you can look at your individual samples and very quickly gain an understanding of how potent the cytokine secretions are from your samples at a single cell level. So looking at why polyfunctional strength index and why characterizing the polyfunctionality phenotype is important, we can look at the fact that certain individual cells are extremely important because they're polyfunctional. So what I mean by that is that you can have polyfunctional cells, and these can be extremely beneficial in some cases, such as in cases where you're looking for polyfunctional cells to kill cancer cells. Polyfunctional cells, the cells capable of secreting multiple cytokines, are able to secrete multiple cytokines in order to elicit a coordinated attack against cancer cells, and thus they have a higher frequency of being able to successfully kill cancer cells and successfully generate good clinical results. There are certain situations too where polyfunctional cells can be considered adverse. For example, in situations where you have a lot of inflammation, different types of autoimmune disease, or organ transplantation. These are all different types of settings where the presence of these polyfunctional cells can be considered bad because these cells are also capable of killing healthy cells. So what our platform really offers is the ability to evaluate these cells and whether to understand if these cells are being beneficial or whether they're being adverse in the cytokine secretions that they're using. And multiple different types of institutions have utilized our platform in order to analyze their cells. I featured a couple of them here on this particular slide. You'll see that our work ranges from preclinical characterization all the way to looking for various types of clinical biomarkers. So looking at the examples presented on this particular page, we're looking at the different types of polyfunctional cells where we really want a lot of polyfunctionality, where we want this cancer killing capability in cancer immunotherapy applications. So you can see that a wide variety of different institutions have utilized our instruments, and they've been able to publish in a lot of different high impact journals, such as Nature Medicine, Nature Communications, Blood, Journal of Clinical Oncology, or Science Translational Medicine. So we'll take a deeper dive into a few of these examples in the coming slide. And again, I just really quickly want to highlight too that our instrument has been applied for a really wide variety of different cell types. So looking at different types of CAR T cells, TCR T cells, TIL cells, CAR NK cells, all of these different cells we're able to characterize using our instrument, and we're able to provide you with a view of cellular heterogeneity and cellular function in the form of the cytokines that are being secreted from these individual cells. So let's start by talking about this particular example here, which was published with Stanford. So one of the things that Stanford was researching was they were looking at this bispecific CAR T that they generated. So the bispecific CAR T was designed to target both CD19 and CD22. Bispecific CAR Ts are important because they allow for each individual CAR T to have more than just one target. So if a cancer tries to evade the CAR T by downregulating expression of, say, CD19, then potentially you can still have the CD22 target available for that bispecific CAR T. However, what they ended up noticing was when they started rolling out this particular therapeutic in the clinic was that they saw a really high prevalence of relapse in these particular patients that were receiving this bispecific CAR T. And the question became, why exactly are these patients really relapsing? This particular therapy should be better in the sense that you have more targets in order for the CAR T to attack. But for some reason, these patients were experiencing relapse. And the particular experiment that they set up here was they wanted to compare the bispecific CAR T to a monospecific CAR T that was designed to just target one of the targets, namely CD22. So when they did this evaluation on our platform, they were able to measure the polyfunctional strength index that I mentioned before of the bispecific CAR T and compare it to the monospecific CAR T. And what they were able to very quickly see was that the monospecific CAR T was capable of higher levels of cytokine secretion through this polyfunctional strength index metric than the bispecific CAR T. So you see here the when stimulated with CD22, the monospecific CAR T had high levels of PSI for both CD4 and CD8 populations compared to the bispecific CAR T that had lower levels of polyfunctional strength index for both the CD4 and CD8 populations. What this really revealed was that even though the bispecific CAR T was designed to attack more targets, the affinity of this particular CAR T seemed to be weaker in the fact that it couldn't mount as many cytokines and it couldn't secrete as many cytokines as a classical monospecific CAR T. So this really revealed that the bispecific was weaker than expected and really shed light on why exactly the clinical translation did not pan out as Stanford has intended. So this provided critical information for them to go back to the drawing board in a sense in order to improve their bispecific CAR T, possibly by changing things such as the affinity, changing things such as the synapse, or changing things such as the co-stimulatory domain in order to improve the polyfunctional strength index of this bispecific CAR T so that it could be more successful in patients. And that's really encapsulated in this reflection statement where it's saying that results suggest that engineering iterations should be guided by careful studies of single-cell CAR polyfunctionality, incorporating cytokine production as a critical quality attribute. So that quote from the Nature Medicine paper really showcases that our platform would have been instrumental for helping the researchers at Stanford understand why exactly this specific bispecific CAR T was not performing up to their standards and potentially reveal some ways as to how exactly they could improve their CAR T in the future. I next want to cover another preclinical study that investigated how exactly cytokine secretions were impacted by various types of CAR designs. So in this particular case, the leaders at St. Jude Research Hospital were investigating CAR NK cells. And specifically, they were researching whether they could improve the synapse binding strength of their CAR NK cells. So they had this particular modification that they made on their CARs in the CAR PDZ zone in order to make their CARs more potent, or at least that's what they theorized. And the reason they turned towards our instrument was they could then use our instrument now to test whether this PDZ modification actually helped to improve the cytokine secretions of their CAR therapeutic. So what they generated was a CAR PDZ, and then they generated various types of controls, such as an untreated CAR, a CAR that was non-functional, or a CAR that was targeting something other than the target cell. Comparing these different types of CAR control constructs to the CAR PDZ construct, you can see that the CAR PDZ had the highest percentage of polyfunctional cells compared to the other controls, and also had the highest polyfunctional strength index compared to the other controls. Furthermore, our technology also allows you to take a deeper dive into the individual cytokines that are being picked up in order to start to understand which cytokines are being upregulated in your different types of treatments. So for example, here you can see that specific cytokines listed, such as IFN gamma, MIP1 beta, and perforin, were all significantly higher in the PDZ construct compared to the untreated construct. So what this really reveals is that this particular, is that our technology was able to help to evaluate that modifying the PDZ domain of these CARs was able to increase the functionality of these CARs and allow these CAR NKs to have a higher chance of success in vivo and in clinical studies, because it is capable of more cytokine secretions and looking through at what our platform was able to offer. As another quick example of how our instrument was used to evaluate anti-tumor efficacy, we can turn towards this particular study done at UCSD that was published in Cell Stem Cell. So looking through here, what they were investigating was whether the deletion of a particular cis gene in NK cells impacted tumor killing ability and in vivo persistence of engineered cells. So looking at this particular graph, you can see that we're comparing the knockout group of the cis gene to two controls, and you can actually see very apparently that when knocking out the cis gene for these NK cells, you're able to dramatically increase the polyfunctionality and the polyfunctional strength index of these particular NK cells. And looking through at how exactly these researchers really reflected on this information, they were able to notice that deletion of the cis gene in human NK cells led to augmented single-cell polyfunctionality, which was a profile that has been demonstrated to positively contribute to clinical outcome in CAR T therapy. So taking all this together, we were really able to, again, utilize our technology to understand why exactly certain therapies might be more effective, and here evaluate that this particular cis gene modification is likely to increase the potency of different types of therapies that might be used in conjunction with these modified NK cells. And moving on to kind of manufacturing settings, when you're looking at manufacturing your CAR T, CAR NK, or even your TIL cells, a big part of looking at the manufacturing process is what particular types of strategies you can use in order to improve the functionality of your cells. And our technology plays a key role in terms of evaluating whether a manufacturing change can generate the right type of functional profile that you're looking for. So this particular study, published in Nature Communications from Worcester, was able to show that the long-acting IL-7 that they had engineered was able to preserve polyfunctionality in the cells that they were manufacturing. So they were manufacturing different types of CAR T cells, and they had this nice engineered IL-7 that they theorized would help improve the functionality of their cells over time. And sure enough, what they found when they utilized our platform was they were able to measure the polyfunctionality of these cells at day 0, and then they compared at day 14 between a standard non-functional vehicle versus utilizing the modified IL-7 during the manufacturing process. And what they're able to find was that utilizing this modified IL-7 was able to help these cells recover their functionality over time. So typically when you end up manufacturing these cells, if you culture them for long periods of time, they end up reaching parts of senescence, right? They start losing their functional performance. But what you can see here is that our platform shows that the modified IL-7 that they were testing was able to help recover some of that polyfunctionality and some of that strength in order to potentially use that in the manufacturing process and to generate stronger CAR Ts in the long run. And all of those examples that I covered before are more or less in the preclinical space. So testing different types of therapies, whether it be in plate-based in vitro assays or in animal-based in vivo assays. But where our technology reaches further heights is where we are able to apply our technology to test clinical samples in order to generate clinically relevant biomarkers. And this particular study from Kite really showcased that capability extremely well. So what Kite was really interested in was the fact that, of course, when you are performing CAR T therapy, some patients will respond extremely well to the therapy, whereas other patients might not respond or might relapse even after CAR T therapy. And the real question here was why exactly that happens. Why is it that certain patients respond and other patients don't? We know inherently that there is some variability between different types of patients because everyone's immune system is different. Maybe the product generated can also vary because of that and because these are different types of autologous therapies. But the real question was, this happens, but why exactly does it happen? And can we find exactly why this happens? And our technology really played a key role in helping to uncover why exactly this happens in patients. And this particular study and this particular graph shown here in the middle is showcasing that we can utilize our polyfunctional strength index to compare the polyfunctional strength profile of responders versus non-responders. And what we found was that responders had statistically significantly higher levels of polyfunctional strength indexes detected from their CAR T cells compared to non-responders. So the fact that this happens really showcases that the functional secretion of cytokines from these individual cells is a critical biomarker that can really help to distinguish your responder population from your non-responder population. And to kind of compare that to some other traditional techniques, Kite also performed a bulk ELISA for IFN gamma, different types of flow cytometry analysis to look at different ratios of surface markers in their cellular populations. And what they found was that the PSI that was generated from our IsoSpark and IsoLight instrument was the only significant predictor that distinguished responders from non-responders. So you can see here that the p-value here fell well under the 0.05 p-value statistic in order to generate statistically significant data. So PSI was a statistically significant indicator that responders had higher levels of functional strength index than non-responders. In comparison, those other standard techniques with bulk ELISA and flow cytometry all had p-values above 0.2, so they were not able to adequately distinguish between responders and non-responders. And this really colors in parts of the future of where single cell functional analysis can go to. where if you really imagine and extrapolate the use of our technology beyond this one case, you can see it as kind of a predictive way to look for different types of biomarkers in these patients in order to see which patients may be responders or non-responders. Now, this particular study done here was a retrospective analysis. So basically, these patients already received the therapy, but later on down the line, let's picture a few years down the line, there is the potential that we can utilize data such as the data generated from the IsoSpark in order to predict whether a patient will be a responder or a non-responder before we deliver the therapeutics, in order to really try and increase the efficacy of the therapies and increase the percentage of patients that will likely be responders. And then for those non-responders, we can potentially talk about different types of strategies that may be more intensive in order to help those non-responders reach a responder status. So really, our platform was able to generate a lot of extremely exciting data for Kite in order to showcase that our technology could be used as a biomarker tool. Furthermore, our technology does not have to just be used at a single time point. So with regards to immune monitoring is also a really important application for these different types of cancer immunotherapies. So the particular example that I'm showing here was performed and published in Nature Medicine and was performed by our researchers at Moffitt and Duke. And what they were interested in was in tumor infiltrating lymphocyte therapy, so TIL therapy. And what they wanted to evaluate was how exactly does TIL therapy affect patients over time, and how exactly does it affect the polyfunctionality of the patient's cells over time. So what they were able to do was they were able to take cells from their patients at various time points in order to see how exactly their polyfunctionality changed over time. So we start here at day -9, at week -9, which was the baseline measurement. And after that, they applied nivolumab, which is a checkpoint inhibitor, and they did not see very much a change in the polyfunctional strength index of these patients' cells. But then you can see at day 0, where they applied the TIL therapy in these patients, you can see this extremely prominent jump in polyfunctionality detected from these patients. So the polyfunctionality of the cells in these patients really increased over time, after injection of these TIL cells. And then looking over time, they also took samples at 4 weeks and at 12 weeks, and were able to show a level of persistence of polyfunctionality for these TILs. And this kind of showcases that there's also a dynamic element that can be brought with our instruments depending on what types of samples that you have available. So this dynamic element can be used to characterize how this polyfunctionality changes over time. And perhaps, let's say, if you're a researcher interested in improving the persistence of your cells in order to in order to decrease the levels of relapse in patients, then you can potentially utilize our technology to evaluate whether that therapy is acting the way that it should. So by seeing if this therapy is able to keep the polyfunctional strength of these cells high over time, or whether it's not able to do so, and whether that polyfunctionality still decreases at the same rate. So a lot of really cool and interesting types of clinical studies that can be done using our technology for these different types of cancer immunotherapies. And I'd also like to share a pretty interesting quote from Moffitt and Duke that was featured in this publication. So PSI reflects the ability of a T cell to carry out multiple functions and is recognized as a metric for the potency of cell therapy and for efficacy of vaccines. So really showcasing that our technology has been accepted from the scientific community as a valuable biomarker in order to look at potency and look at evaluating these different types of cellular therapies. So I've just went over a lot of examples, and I hope that I've colored in a lot of different types of examples across the cancer immunotherapy development pipeline. So all of the examples that we went over, right, span from the preclinical to the manufacturing stage, all the way to the clinical stage, where our technology has really been applied in a wide variety of different ways in order to provide the researchers that use our technology with greater understanding of how their cells do what they do and what function really drives the different types of outcomes that they're looking for, right? Whether it's being more killing in the preclinical sense, more potency in the manufacturing sense, or better clinical outcomes in clinical translation settings. And before I close off this presentation, I just want to cover some examples too, where there are certain cases where this polyfunctionality is not desirable. So most of the cases that we talked about before were all in cancer immunotherapy, right? So in cancer immunotherapy, you really do want your different types of immune cells to secrete lots of cytokines and kill lots of cancer cells, but uncontrolled cytokine secretion can lead to a wide variety of different side effects and diseases. So I just really quickly want to highlight that our technology can also be used for those different types of applications. So for example, our technology has been used also to evaluate different things such as rejection of different biomaterials, different strategies to improve organ transplantation by decreasing rejection. Also, it's been utilized to look at how immune cells are acting in autoimmune disease and how they're also acting in different types of infectious diseases. So these are some of the examples of our technology being applied in those cases. And again, you can see a lot of really great institutions have utilized our technology, and we've been featured in a lot of very high impact journals. And I'll just cover one example where particular cytokine secretions were investigated and were found to be detrimental to different types of clinical outcomes. And namely here, we are still looking at cancer, so follicular lymphoma here. And what these researchers at the Mayo Clinic were investigating was these particular types of SRIP alpha cells that may have been correlated with worse results in cancer patients. So what they were able to do was they assayed these SIRP alpha high cells, and they were able to see that the cells that had this that were in this particular category had high levels of various types of cytokine secretions, such as IL-10, MCP-1, and MIF and MIP-1 alpha. So looking at different types of cytokines, right, that these cells are secreting, they saw that these cytokines were likely drivers for the poorer results that they saw. And also, apologies, I'll also note that they were looking here at monocytes. So this particular monocyte population seemed to almost be enacting a level of immune suppression on these cells with these cytokines. So again, these cytokines are not always necessarily positive. Sometimes they can also have suppressive effects if we're looking in cancer environments. And then the suppressive environment may be decreasing the actual efficacy of other types of immunotherapies. So just looking at what this paper really offers, functional phenotyping identified the specific novel monocyte that had the functional profile driving immune suppression in these cancer patients. And as next steps, potentially identified some strategies in order to decrease levels of these cytokines and hopefully then improve the outcomes from patients that have higher levels of this particular type of cell. And with that, to summarize, we've went over the Light and IsoSpark single cell secretome platform. So again, using our chip, you're evaluating multiple different cytokines being secreted from these individual cells. You're measuring 30 plus cytokines, and you're getting a lot more depth of information than you can gain with traditional technologies such as flow cytometry or bulk ELISA. And our technology really specializes at giving you that single cell perspective of functionality of what these cells are actually doing and what these cells are actually enacting on other cells, such as cancer cells. And with that, to summarize, again, we've went over the single cell secretome platform, but we do have other platforms, and I would encourage you to either reach out to a representative at Bruker Cellular Analysis or, of course, our local distributor, Rhino Bio, in order to learn more if you would like. And our other platforms are, again, a bulk analysis platform that allows you to look at different types of proteins in biofluids. So if you do want a quick look at your different types of cell supernate or plasma, you can utilize our bulk platform to provide you with that information. And also our intracellular signaling platform, where we're able to look at the individual phosphoproteins from these single cells and looking at, again, at a single cell level, what phosphoproteins are being upregulated in certain cases or downregulated. And with that, just want to conclude the talk. And again, a quick thank you to Rhino Bio for organizing this webinar and for being our local distributor in the Korea region. Happy to address any questions, and thank you again for your attendance and your attention.